All right, our next speaker is Nikola Knoll from Georgetown University, speaking on PAX3 FOXO1-targeting PROTACs in fusion-positive rhabdomyosarcoma. Welcome.
Right, good morning. I'm Nikola. I'm a second-year PhD student in the URAN lab at Georgetown University. And it's my pleasure to talk about our PAX3 FOXO1-targeting PROTACs in fusion-positive rhabdomyosarcoma. So just a few words about rhabdomyosarcoma, or short RMS. It's a tumor that occurs usually in or around skeletal muscle, and one specific subtype of rhabdomyosarcoma has these chromosomal translocations, which gives rise to these fusion proteins. So these are uniquely expressed in the tumors. And the most common of which is PAX3 FOXO1. And this fusion protein is driving the disease. It's the defining feature of fusion-positive rhabdomyosarcoma. And fusion genes are occurring when two genes that are usually separated from each other are now fused together in the tumor, giving rise to this novel, unique protein in the tumor cells. And PAX3 FOXO1, as I mentioned, is the defining feature of fusion-positive rhabdomyosarcoma. And fusion-positive rhabdomyosarcoma is unfortunately a lethal disease. So less than 30% of these patients survive longer than 5 years. So there's a clear unmet clinical need to develop better treatment. And given PAX3 FOXO1's unique expression as well as its oncogenic role, it represents an attractive target. Our goal is to eliminate this disease-causing protein to stop fusion-positive rhabdomyosarcoma growth. So why don't we already have a treatment? Well, it's difficult since PAX3 FOXO1 is a transcription factor. And if you think about conventional small molecule inhibitors, they require those deep enzymatic, often pockets, to bind and inhibit the activity of the protein. But if you look at the predicted structure of PAX3 FOXO1, there are large segments of intrinsic disorder. That means they don't have a defined structure, and there's no deep binding pocket to which a small molecule inhibitor can be easily designed. So with that, we turn to targeted protein degradation, which takes advantage of the cell's own ubiquitin proteasome system. You can think of it as like the cell garbage disposal system. So you have a protein, the substrate in the cells, and then it binds to this E3 ligase, which is then adding ubiquitin molecules on this protein, which is a signal for the cells that this protein is no longer needed, and it is sent to the proteasome, which is then degrading, meaning completely eliminating this protein from the cells. So now we want to do the same with PAX3 FOXO1. However, in normal cells, PAX3 FOXO1 might not always bind to this E3 ligase, meaning it cannot be polyubiquitinated. So we're using these PROTAC molecules, which have 3 main parts. One part is the E3 ligase ligand, which is binding to that E3 ligase. The other side of the molecule is binding to the target protein, which is PAX3 FOXO1, and then there's a small linker. So when we add PROTAC to the cells, it brings the E3 ligase and the PAX3 FOXO1 protein into close proximity, which is then allowing the E3 ligase to polyubiquitinate PAX3 FOXO1, so adding these ubiquitin molecules, which is the signal for the cell to eliminate it. So it's sent to the proteasome and is degraded. So the main advantages of this system is it's eliminating a protein, but also these PROTAC molecules don't require this high affinity binding to the target as would small molecule inhibitors. Even weak affinity interactions are sufficient to degrade the protein. So with that, we want to design PAX3 FOXO1 targeting PROTACs, and the first thing what we need for that is a binder to the target. So a compound that is binding directly to PAX3 FOXO1 protein. And to do that, we used surface plasmon resonance technology, which is technology that is very suitable to screen small molecules for direct interaction with, in our case, PAX3 FOXO1. So we screened these molecules for direct interaction. This was previously published by the lab, and from there we identified several molecules that directly bound to PAX3 FOXO1 protein. And we used these binders for our PROTAC design. So if we look here again, we see those 3 components of the PROTAC molecule. We have on one side the E3 ligase ligand. In our case, we started with a cereblon ligand just because it's the most commonly used E3 ligase in PROTAC design. used a variety of linkers, and then the PAXI-FOXO1 binders that we identified from the screen. So with these initially 15 PAXI-FOXO1 targeting PROTACs that we called PFPs, we tested them in fusion-positive rhabdomyosarcoma cells. And if we compare it to the DMSO untreated control, we can see that with our treatment, we can actually reduce PAXI-FOXO1 protein levels, meaning we are degrading it. So we're kind of happy with that it worked initially, but still the potency wasn't good enough. So we thought, how could we further optimize this? And one way of doing this was to switch to E3 ligase. So we thought if we look at expression data of E3 ligases in fusion-positive rhabdomyosarcoma cell lines, as well as patient tumors, we see that cereblon is kind of expressed on an average expression. However, there are several E3 ligases that are just simply higher expressed. So we thought that's a good starting point. So we looked at SCIP1, which is really highly expressed in both tumors and cell lines, and also validated this in our cells, where we see a 20-fold higher expression of SCIP1 compared to cereblon. Next, we looked at dependency data, meaning whether an E3 ligase is essential for the tumor cells to survive. So cereblon, for example, is not essential. So cells can simply downregulate the expression to prevent the mechanism of the PROTAC molecule, meaning resistance mechanisms. However, SCIP1 is a strong dependency in these fusion-positive rhabdomyosarcoma cells, meaning they cannot survive without it. These PROTAC molecules now using SCIP1 are less prone to resistance mechanisms. And lastly, we also found a SCIP1 recruiter that we could incorporate into our PROTAC design. So this is the new molecule that we designed now using this SCIP1 recruiter, linker, and then a part that is binding to the PAXI-FOXO1 protein. And now we compared them side by side, the SCIP1-based PROTAC that we call PFP16, and compared it to the cereblon-based counterpart, PFP4. And we can see that PFP16 was much more potently degrading PAXI-FOXO1 protein in fusion-positive rhabdomyosarcoma cells compared to the cereblon-based PROTAC molecule. So, for example, the half maximal degradation coefficient for PFP16 was around 8 micromolar, and the same concentration, 8 micromolar, only achieved around 25% degradation for the cereblon-based counterpart. So next we wanted to test whether our PROTACs are actually degrading PAXI-FOXO1 protein in the mechanism that the molecule is designed to do so, meaning by polyubiquitination of PAXI-FOXO1 protein and subsequent proteasomal degradation. So first we looked at polyubiquitination. We used co-immunoprecipitation, so pull down of PAXI-FOXO1, and then plotted for ubiquitin. And we can see in the untreated DMSO control, we see no signal of DMSO. However, when we add the PROTAC molecule, we see a strong increase in ubiquitin signal, indicating multiple ubiquitin molecules were added to the PAXI-FOXO1 protein. Next, we looked at the proteasome. So we used the proteasome inhibitor, MG132, which is just inhibiting the proteasome, and without the proteasome, we couldn't degrade PAXI-FOXO1 anymore. So we stabilized PAXI-FOXO1 in the presence of this inhibitor. And similarly, we also see the same trend with MLN4924, which is a NET8 activating enzyme inhibitor. And this is a required step for the activation of the SCIP1 E3 ligase. So by inhibiting the activity of SCIP1, we could also stabilize PAXI-FOXO1 protein. So these experiments told us that we are indeed polyubiquitinating PAXI-FOXO1 with an active E3 ligase, and this degradation then is performed by the proteasome. So now that we have a PROTAC molecule that is degrading PAXI-FOXO1 in a concentration and proteasome-dependent manner, we're wondering what the actual biological significance of this is in fusion-positive rhabdomyosarcoma cells. So as a transcription factor, PAXI-FOXO1 has several genes that it's regulating. So there are several target genes that are described in the literature. So we compared the expression profile of our PROTAC-treated cells to those known target genes. And we do see an enrichment of these genes in our PROTAC-treated cells, meaning that we are deregulating PAXI-FOXO1 target gene expression. Next, we also looked at hallmark gene sets and saw an enrichment in myogenesis in PROTAC-treated cells, meaning an increase in expression of genes that are important for myogenesis, as well as a downregulation of MYC targets or ETF targets, So overall, our PROTAC treatment was able to induce myogenesis and downregulating FOXO1-related pathways in fusion-positive rhabdomyosarcoma cells. So a few more words about myogenic differentiation. I mentioned rhabdomyosarcoma is a tumor of the skeletal muscle. However, FOXO1 is able to prevent the differentiation of these tumor cells into muscle cells, meaning it's keeping them in this undifferentiated state. So by inhibiting FOXO1 function, other labs have shown that these tumor cells can differentiate into muscle cells. So in our case, we're degrading FOXO1, so we're hypothesizing that we also are inducing myogenic differentiation. And muscle cells are expressing specific markers, such as myogenin or mucin heavy chain 3. And by using our PROTAC treatment, we could increase the expression of these genes in fusion-positive cells, but not in fusion-negative RD cells that simply don't have FOXO1 protein. And similarly, we see this on a protein level, where we see a dose-dependent degradation of FOXO1 protein, with that a decrease in MUD1 levels, which is a target gene of FOXO1, but at the same time an increase in MUG levels, which is mu differentiation marker. We further validated myogenic differentiation with immunofluorescence staining. So in green, we're staining for mucin heavy chain, the mu differentiation marker. So in the untreated control cells of ARCH41, we don't see mucin heavy chain signal, but then with the PROTAC treatment, we induce a strong expression of this muscle-specific gene. And we also see this more spindle-like morphology, indicating more muscle-like cells. We don't see this in the fusion-negative cells. And next, we also looked whether we can sensitize these tumor cells to vincristine treatment. Vincristine is one of the chemotherapeutics that is commonly used for one of the standard of care treatments in rhabdomyosarcoma. And as we just heard, reducing any dose of any toxic chemotherapeutic is really important. So with our PROTAC treatment, we could sensitize these cells more than 100-fold to vincristine treatment, meaning less of this toxic chemotherapeutic is needed to kill these tumor cells. We also tested this across a concentration range and used this Bliss synergy model to calculate a score, indicating that these two treatments are potentiating their effects in killing these tumor cells. And lastly, we also tested anchorage-independent growth in soft agar of these tumor cells, where we can see that small colonies are forming in this 3D soft agar under untreated conditions, but then with increasing concentrations of our PROTAC treatment, we are able to impair their growth, to inhibit this tumor cell growth. And we don't see this in an unrelated cell line, TC71, which is a Ewing sarcoma cell line, which don't have FOXO1, showing that this is a fusion-dependent mechanism. So just to quickly summarize, we used SBR to identify probes that we then used for the design of PROTACs. We designed FOXO1-targeting PROTACs that were able to degrade FOXO1 in fusion-positive rhabdomyosarcoma cells. These PROTACs were degrading FOXO1 via ubiquitination and proteasomal degradation, and were able to impair FOXO1 target gene expression and induce myogenic differentiation. And importantly, FOXO1-targeting PROTACs are impairing anchorage-independent growth of these tumor cells and synergize with vincristine treatment in fusion-positive rhabdomyosarcoma cells. So in the future, we hope to further optimize these PROTAC molecules. We also want to investigate potential off-target effects and, most importantly, determine the therapeutic potential of these molecules. And with that, the most important slide, I want to thank everybody, especially Dr. Oren, who is a great mentor and guided me through this project, everybody in the lab, as well as the Turetsky lab, who are great to work with, and also the funding sources, especially CCF. Without them, none of this would be possible.
So in thinking about PROTACs, right, FOXO is lost in the protein. But if I understood what you thought correctly, it wasn't lost. So the question is, how much do you have to treat efficacy? If you have residual FOXO protein, is that enough? And then if I can ask a second question,
Yeah, I think that's a great point. I mean, these are early candidate molecules, so they still need a big amount of optimization. We're still in the micromolar range, so yeah, and we were not able to completely degrade FOXO1 protein. However, I do have to say that with the doses that we did use, we did see this significant impairment of anchorage-independent growth, even at doses that were not able to fully degrade the protein. So we do think that there is some therapeutic window, but yeah, there's definitely more work to be done.
Do you have any sense about how to develop acquired?
That's another great point. I slightly touched on it. So for cereblon, which is already also in clinical trials, there's also one PROTAC molecule that has been approved by the FDA just recently. And for especially cereblon-based PROTACs, there are resistance mechanisms involving just the downregulation of the E3 ligase. So cereblon is just not expressed anymore. And this is possible because cereblon is not essential for these tumor cells. So that's why we looked at SCIP1, which is a common essential gene. So tumor cells would have to find a different way of getting rid of this PROTAC molecule. Because SCIP1 expression is required for these cells.
Silence SCIP1 in the cells to see that you lost the activity?
That's a great point. We haven't done that experiment simply because SCIP1 is essential. So other people have tried, and in tumor cells especially, you don't get to the point where SCIP1 is not expressed, and then you can test efficacy because these cells don't survive that. But yeah, I haven't given it a lot of tries, to be honest. Yes. Yes, of course, yeah. So SCIP1 is the core requirement for this SCF complex. And then SCIP2 is one of the FBOX receptors. So this one is interchangeable with other FBOX receptors. And we don't currently know which FBOX receptor is needed for this PROTAC mechanism, but it's also a great point.
Yeah, thank you, Nikola. Very nice talk over here in the middle, right in front of you. This is a really tough project, and congratulations for tackling it. Just curious about the specificity of your binders, and do you know if they bind, where do they bind to the fusion protein? Do they form a ternary complex? Just thinking about the high micromolar concentrations that you're needing right now to see some degradation.
Yeah, these are excellent points, and unfortunately the short answer is we don't know yet. We're working on these things. The ternary complex formation has been a bit difficult to easily test since the SCIP1 recruiter that we're using, it was described by the Nomura group, and they show that it's only binding in their interface with an FBOX receptor protein. So you need this whole complex to then do those studies. So we're still working on this, so we don't currently have an answer to that. And regarding the specificity, that's also ongoing. We don't yet have the selectivity profile. Most likely there are other things that are probably targeted in some way or another by this PROTAC molecule. That's also ongoing. And then the last part, where it's binding, again, a great question. We don't know yet. Those structure-based screens are not really feasible since it's so disordered. So we don't really get a high confidence prediction from these tools. We believe it binds on the PAX3 side since we saw some kind of degradation there, but then there's not a great PAX3 antibody that we can use. So there are several things we're still working on, but we don't know yet.